Welcome everybody. So in this episode of the Blue Brain seminar series, today we have Alessio Buccino visiting us. I mean, actually, Alessio was already. Yeah, you can come here. Actually, Alessio was already here in 2019 also for a young seminar series, a young scientist seminar series. And I mean, at that point, we actually were discussing like how we could collaborate, maybe on how we can use extracellular recordings to constrain our models and put that in our BluePyOp software. So now we are 3 years later, and Alessio is back, and so we're basically finalizing this collaboration now, and finalizing the paper on this topic. And so, yeah, I mean, Alessio will give us, show us the progress of our work, basically. So Alessio has been started his PhD in Oslo with Gata Einevold on working on with the LFI software, which you will actually see in this talk. Then afterwards, he came to Switzerland, worked at the ETH for Andreas Hilleman. So that's basically part of the things he did there was this work. And now Alessio is working as a almost full-time consultant for the Ellen Blaine Institute for Neurodynamics, and also, you also told me that you're 10% consultancy, I think, for a company that like helps scientists, like for example, converting their data. So welcome, Alessio, and go ahead with you.
Hello, everyone here and remotely. So this means that now this is a senior seminar series. I made it. Yeah, so today I'm going to be talking about basically what Werner introduced. So this was my main postdoctoral project, and it's about a multimodal fitting strategy or approach to construct single neuron models by combining patch clamp and high-density microelectrode arrays techniques. So let me start. As Werner introduced, this was like a collaboration between two large Swiss institutions. And we were recalling before that as soon as I joined the ETH for my postdoctoral fellowship, and it was in March 2020, so right at the start of the pandemic, I reached out to Werner saying, hey, let's do this project together as we discussed before. And actually, I'm very happy to be here. That means that it worked. And I really enjoyed this collaboration. And I want to actually thank everyone who participated. We're kind of like half and half between ETH and the Blue Brain project at EPFL. We did most of the experimental part, and you at the Blue Brain project, most of the modeling. I would try to glue these two things together. And so I hope you enjoy what I'm going to present. So I'm going to have four main sections in this presentation. So we'll start with a very brief introduction to multi-compartment models, and then we'll see what we need to do in order to use multiple techniques, so a multimodal strategy to fit single neuron models to the data. And then I'll show how we evaluated this technique and this approach using some ground truth models with known parameters. And finally, I will show some experimental data and some results that we obtained on data from cell cultures. So let's start with the multi-compartment models. Most of you are probably familiar with, but if someone is not familiar, multi-compartment models are among the most biophysically detailed models that can represent the single neurons in terms of their full dynamics, and they're mainly constructed of two different parts. So the first part is the morphology, and this is usually extracted from high resolution imaging, or also like at different depth levels, the so-called Z-stacks, from where we can actually trace and reconstruct a precise geometry of the neuron in 3D, with also the diameters of the different neurites, what are the different neurites, and so on. So this is very detailed. And then on top of this, we have to populate this morphology, bare morphology, with some electrical properties. And in order to do that, we need to use some experimental data. The gold standard is using a patch clamp data. We see here a trace. And then basically we try to fit the many parameters that these models are made of to the experimental data that we basically acquired to find a model that fits them well. I want to say that basically in the last few years, maybe now it's been 10 years or more, there's been actually a lot of international effort in order to actually construct these models and also to share them publicly in different online databases. For example, of course, the Blueprint projects at their reconstruction of the neocortical microcircuits, and they're always working on building more models from different brain regions, different species, and so on. And a similar effort is also carried out by the Allen Institute of Brain Science, and they have several models, mainly from visual cortex, from mice, and even from humans. So these models can really help research in terms of trying to test hypotheses first in the models or try to drive new experiments and vice versa. But we actually identified a main problematic for these models. So you can see here the 3D structure of a neuron is really like elongated and heterogeneous, but most of the data that is acquired and fitted to construct these models is only from the soma, right? So how could you say something of what's happening like very far from the soma? These are can be several, like a couple of millimeters in humans, just by looking at one recording from very far away, right? Neurons are more than just the soma. They have like complex dynamics also in these other neurites, in the dendrites, the axon initial segment. The axon itself is particularly also thought to be carrying information, right? So maybe just looking at the soma might be a limiting factor. And in fact, there are some previous studies that try to use other techniques in order to better fit basically other neurites more in a more detailed way. For example, in this Hay model from 2011, they didn't use a single patch recording, but 3 simultaneous recordings, so one from the soma and 2 from the apical dendrites. And they actually showed that these provide a much better model in terms of fitting the dendritic dynamics, right? But on the other hand, having multiple patch pipettes recording the same neurons is very, very experimentally challenging. Even recording one neuron with the patch clamp is experimentally challenging. Even more, like the same neuron with more pipettes, it's really complicated. So we try to find basically an alternative solution using multiple modalities, so not only patch clamp. And in particular, I joined the lab of Andreas Hierlemann, as Werner said, in ETH Zurich, but the lab is in Basel. And they have developed over the years a recording system that is based on high density microelectrode arrays. So basically, these are schematic of how it looks. So basically, you have many small electrodes. These are actually in the order of 15 micrometers apart from each other, and neurons sit on top of them. So you can really record a spatially distributed information of the entire neuron. You can see here just a movie of like one single average action potential from a neuron, and you see how rich the spatial information is, right? So this kind of like approach combined with patch clamp, which instead gives very precise, both temporally and spatially, and it also allows us to stimulate the neuron, could actually be a good way to construct multi-compartment models. But before diving into this new approach, I would like to guide you through what's the common way to build these models just using patch clamp, right? So the workflow, of course, starts with data acquisition. So we identify a target cell that we want to construct a model for. We try to patch it. Maybe after several trials, we can actually patch that cell or another cell close by. And then we apply some protocols, which are mainly some current stimulus, some steps, or some more complicated stimulation protocols, and we record the responses of the neuron. After the experiment is done, since the patch pipette can be filled with some fluorescent dyes or some like high contrast dyes, we can reconstruct the precise morphology of the neuron. So these are kind of the 2 input data that we need to the model. Then what we can do is basically from the responses that are high dimensional, so you could think that these are like time series that could be seconds or even sometimes minutes long, depending on how many protocols you have. So we need to kind of like reduce the dimensionality so we can extract some features from these responses. Like, for example, what's the action potential amplitude? What's the time to the first spike? What's the average firing rate in this particular protocol, and so on. And then we also have to define what the models look like. So what are the mechanisms, or let's say the conductance model, the conductance-based models of this cell? So where are the sodium channels? Where are the calcium? What is the calcium dynamics? What kind of potassium channels is this cell basically expressing? And this is mainly the task of the modeler, to know what kind of cell, based on literature and on previous experiment, how to distribute these mechanisms across the cell, right? And then that's the fun part, is the optimization. So basically what we do is we combine all this information with a simulator to build like an in-silico replica of the experiment, right? From this simulation, we then extract the same feature, and then we iteratively explore the parameter space to evaluate how well a certain solution is fitting the experimental data. We do this basically iteratively, usually using genetic algorithm, and we use the software developed by the Blueprint project called BluePyOpt. We try to fit different objectives for different features, but the bottom line is that after some time, we find a model that's actually optimizing our data, and we say that this is the fitted model that we're going to use. Now, how do we have to extend this pipeline in order to use extracellular data as well? Well, first of all, of course, we need to extend the data acquisition part. Well, before we only needed patch clamp, right? Now we need patch clamp and extracellular recording simultaneously, right? Second, we also need different set of features, right? There are commonly used features that have been defined and used for patch clamp traces. Now we have extracellular traces, so we need to come up with a new set of features somehow. And then at the optimization side, we need to change the simulator. So we need to use a simulator that also allow us to reproduce these extracellular signals on top of the intracellular traces. So let's start with the data acquisition. So we performed experiments in which we had the cell sitting on top of the high density microelectrode array, and then we patched the cell, and we can see here an example of the simultaneously recorded intra and extracellular traces. So you can see that actually for each intracellular spike, we can observe the neuron also extracellularly on multiple electrodes because they're so close by. And then we used a technique which is called patch-triggered average, or maybe we called it that way. So basically, we use the spikes detected from the intracellular trace, and we average the extracellular signals to extract an average extracellular template. And we do so because you might know that extracellular signals are much noisier than intracellular ones. So the signal to noise ratio is much lower. So we need to actually perform this averaging to be able to observe, for example, small signals coming from the dendrites or from the axon, because they would be just buried in noise if we just observe them in the recording. Then we need to extend the simulation part. And in order to do so, we added a new backend simulator to the BluePyOpt software, which is called LFPy. So LFPy is a Python package that has been developed by Gaute Einbol and Espen Hagen at the University of Oslo. And basically, it's a wrapper on top of the neuron simulator that also allows to compute extracellular signals. So the idea is that, as you may be familiar with, the neuron simulator allows you to solve the cable equation. So, right, to get the intracellular dynamics of a neuron. And part from this equation, once it's solved, we know the transmembrane currents that are crossing each of the, let's say, compartment at each time, right? And then we can use volume conduction theory to actually compute from these transmembrane currents what would be the extracellular potential at recording electrode position, right? Using these relatively simple formulas. Once we have these extracellular signals, as I said before, we also need to come up with a different set of features, right? And we looked in the literature and also tried to define some new features. And basically here we see an example of an experimentally recorded extracellular template. And here I'm just highlighting the signal on the channel with the largest amplitude and another channel. This would be close to a dendrite. So basically we define two sets of features. One we call absolute features because they are computed for each channel separately, right? And some of these are, for example, what's the peak to trough ratio, what's the half width, what is the peak to valley duration, the recovery slope, prepolarization slope, and so on. And then we have another set of four features, which instead are, we call relative features. So they are computed with respect to the channel with the largest amplitude, right? For example, we can say, what is the distribution of the negative peaks, right? So what is the relative amplitude for each channel with respect to the negative amplitude of the maximum channel? And we can do this also with timing. When does the negative peak on any channel occur with respect to the negative peak on the maximum channel? So we get these basically spatially distributed features for all of these new, let's say, 11 features that we defined. Now, there's still a problem, though, that these extracellular feature maps, differently from intracellular features, are multidimensional, right? An intracellular feature, for example, the action potential amplitude, let's say, is like 55 millivolts. It's one value, right? Instead, these features are actually defined on several recording electrodes. This is in the hundreds, right? For these cells, we have recorded, let's say, 300-400 electrodes or even more, right? So how do we come up with single values from this multidimensional feature? So we actually tried three different strategies also to mix the balance between intracellular and extracellular features. So the first strategy, we called it single. So the idea is that we selected manually for each cell a bunch of electrodes, like 6 or 7, and then we consider each of these features. Now it's a scalar, right? So we can compute the objective as we did basically for normal intracellular features, just by comparing the value that we get in the simulation to the value that we get from the experimental data. Then we define another strategy, which is called all. So this strategy actually tries to use the entire information that is there. And in order to get one scalar value out of it, we compute the cosine distance between the vector of the features from the experiments and the vector from the feature from the simulation, right? If we compute the cosine distance, this will give us just one scalar. So this strategy adds to the simulation 11 new features, which are the number of features that we defined. And then we have kind of a hybrid strategy, which is called sections. So in this case, we manually draw a number of subsections that could be informative for the neuron, for example, the perisomatic area, a section close to the dendrites, one close to the axon, and so on. And then for these different sections, we compute the cosine distance, right? So in this case, we have number of features times number of sections, additional extracellular features. I forgot to mention for the single one, of course, we have the number of features times the number of channels that we selected as extracellular additional features. So now that we have extended our methodology, we have to evaluate it, right? In order to evaluate it, we decided to start from a model that we call ground truth, from which we actually know all the parameters. So we know how to quantify how the different strategies are performing. Oops. So we start from the morphology from the Hay model. Okay, this should be 2011. Sorry about that. And the Hay model is the one I basically explained before with multiple pipettes. It has a rich dendritic complexity, but one thing that it doesn't capture, and it's actually quite important, is the dynamics of the axon initial segment, or AIS. Actually, there's a large body of evidence showing that the axon initial segment is where the action potential initiates. and also it's where you have the strongest currents. So this strongly influences the extracellular landscape that you're recording. So that usually, basically, when you look at the extracellular signals, which this paper is from Buccino and colleagues from 2018, the largest signal is observed in proximity of the axon initial segment, not in proximity of the soma, just because of these strong transmembrane currents. So what we did is basically to take the Hay model morphology and then to construct a new model with an additional AIS in there. And the AIS basically was the first tract of the axon. You can see it here in green. And there we expressed AIS specific mechanisms like NIV 1.2, 1.6, and like potassium KV channels. And we saw if actually these new AIS enriched pyramidal cell can actually reproduce features that we observe that are basically due to the AIS. And in fact, it does. If we see here the intracellular signals, the action potential is actually initiated at the AIS, so not at the soma. And then if we look at the extracellular signal, here this is a simulated extracellular multi-electrode array with 80 channels, as you can see here. The channel or electrode with the largest amplitude is actually in proximity of the AIS, right? So this model successfully does what we designed it for. So it has this additional AIS dynamics. So now with this ground truth model, we can start asking the question, does including extracellular features and this multimodal strategy improve the overall quality of the model, right? Do we get better models out of this? So what we did with this ground truth model is to replicate the entire experimental scenario that we also were doing in parallel with experiments. So we used a set of protocols which are called ECODE, and I guess some of you might be familiar with. So they include many different protocol types to basically probe what's the firing pattern of the cell, what are the subthreshold responses, are they bursting or not, what's the precise, let's say, shape of the waveform, and also some more complex protocols that actually look at the depolarization after like a hyperpolarizing pulse. And then finally we have these post Cheops, which is called like this because it reminds of the pyramids of Cheops, I guess. Okay, so we extracted features from this model and we ran these different optimization strategies, and here are the results. So what I'm showing here is the optimization result using 10 different random seeds, right? So basically 10 separate optimizations for each strategy. And on the left, we show the fitness or the total score of the intracellular features that we had. And on the right is the total score of the extracellular features that we had, right? So we can see here that basically the blue is the soma, and then the orange and green are the all and section strategy. And we can see that in terms of intracellular fitness, they seem on par. Actually, the all strategy, so using all electrodes at once, seems even a little bit better than using soma alone. And this is looking at intracellular features, right? The single one, instead, it looks a little bit off. So it has a higher score. That score, in this case, it means error, right? So the lower the score, the better, just to be clear. But when we look at the extracellular features, actually all strategies that somehow used the extracellular information perform better than soma, maybe expectedly, right? But then we could say that especially for all end sections, we get better fitness, right? Because we have a similar, if not better, intracellular fit and a better extracellular fit. But another thing that's interesting to observe is that this kind of like behavior of the single, right? It could reflect the balancing between intracellular and extracellular features that we see in this table here. So for the single strategy, we have almost as many intracellular features as extracellular features. So it might be that basically the optimization is kind of biased towards fitting the extracellular features too much. So it kind of like forgets about the intracellular features. While the all and sections, it might be a better balance in terms of like combining these features from these two modalities. So after we've seen the overall optimization performance, now we focused on one single solution, so the best model. And the best model, for the best model, we used the one that yielded the best intracellular score. So if we look now, we basically, it's only one solution, but instead of providing a single score, we plot here the distribution of all the features that we used for intracellular, from the intracellular traces. And we actually found no significant difference, right, between all these strategies, meaning that all four strategies on this ground truth model are perfectly capable, as you can see also from the traces, of reproducing the intracellular behavior. But then when we look at the extracellular behavior, here is the templates are actually normalized for each channel, so we can see even small differences that happen, for example, close to the dendrites and so on. The strategies that used extracellular information do much better, significantly better than the somatic strategy. I mean, we can see it here from these box plots, but we can also just see it visually here. There are actually large variation, let's say relative variations, especially moving away from the soma. So our initial hypothesis that the soma might not be, just using patch clamp might not be ideal to fit the entire dynamics of the cell, might actually be a good assumption. Okay, after we have validated our model, so we have showed that if everything is ideal, so if we have a model that basically we know the right mechanisms, if we know how to simulate the extracellular signals correctly, then using these extracellular signals for the model fitting is actually giving us a better model, right? We tried actually also to apply this strategy on experimental data. So let's see basically first, how did the experimental procedure, how was it like carried out? So we used data from cell cultures. So cell cultures from embryonic rats. So we extracted embryonic neurons from rats embryos. And then we, after some lab work, we plate them on the MEA chip and we let them grow there. So we let them grow for 2-3 weeks, even a little bit more. And then after, let's say, 18 to 20 days, we performed the experiment. On the day of the experiment, we took the chip with the cells on top on our patch MEA rig. We performed the simultaneous recording of the patch clamp and the MEA with the patch clamp and MEA system. From these, we get the MEA and patch data as raw data, and then we can process these and get the intra and extracellular features that we use for fitting. On the same day of the experiment, since we actually have some fluorescence dye in the pipette, then we use a confocal microscope to perform a Z-stack of the cell. And this is used later on to reconstruct the three-dimensional morphology of the neuron. And then we fixate the culture with PFA so that after a couple of weeks, we can stain specifically for the AIS. Because as we said, the AIS seems to have a very strong influence on the extracellular signals and also on the intracellular dynamics. So then we image the stained culture. We try to colocalize the AIS to the morphological reconstruction that we did before so that we can add the AIS to our morphology. So let me show in more detail how some of these data look, right? So this is an example of a recording, a simultaneous recording of one cell. And in orange here, you have, these are the fire pattern protocols with these two protocols with increasing current. So you can see the firing rate increases. And then you can nicely see one extracellular recording, so this is a filtered extracellular trace, how every spikes perfectly match also extracellularly. So then using this patch triggered average that I introduced before, we can compute this nice and clean extracellular template or footprint. We call it footprint because you can really see, if you just look, for example, in this case is the maximum amplitude of each electrode, you can really try to see the actual neuron morphology from these extracellular signals. It's really an imaging technique, if you may. For the 3D model reconstruction, we used an open source software, which is built in Fiji, which is called Simple Neural Tracer. So these are semi-automatic reconstruction tools. Basically, it allows you to start a path and then select some points along the path, and then it automatically finds the paths and fits the diameters as well. So you can see here how the reconstruction perfectly overlaps with the image. And this is actually our three-dimensional reconstruction model of the cell. And this is one example of the AIS staining. So we stained for two different AIS specific markers. One is ankyrin G, and the other one is the KV7, which are both. So ankyrin G is the structural protein that is expressed in the AIS, and KV7 is an ion channel that is specifically expressed in the AIS. So here basically is the one cell that has been filled with the dye from the patch clamp, which is Alexa fluor. Oops. Here we see the staining for ankyrin G, and then we can clearly see that this neurite over here, right, is actually perfectly colocalizes with the AIS. So then we can say in the morphology, that part of the neuron is our AIS. So we use data and we tried to build model from data for two different cells that were recorded between DIV, which stands for days in vitro, 18 and 24. This is just an overview of the kind of data that we have, and we also shared this data in Neurodata Without Borders format, and it's openly available if you want to have a look. So this is how the cell looks like. We call them internally, we used to call them beauties, because they're just so nice. And then this, the morphology reconstruction overlapped with the extracellular template. In green here, you have the AIS, and in blue is the nice axonal branching. Then here we see the AIS staining. In this case, we can clearly see the AIS starts from here. It's very common for cells from cell culture that their AIS doesn't start directly from the soma, but it actually starts from the dendrite, which is called this axon-bearing dendrite. And then this is just an example of some of the somatic traces. So this is experimental data. Some of the somatic traces that we acquired from this cell, the AP waveform, some fire pattern, these post-Cheops, the cell fires like crazy. And so this was the first cell we had. This is the second cell. You can see the morphology is a bit different, but also in this case, you have this massive axonal arbor that we are able to track. And then this, the overlapping of the morphology and the extracellular template. In this case, the AAS was a bit more faint, but actually you can see that it overlaps here with ankyrin G. And this is an example of the somatic traces. You can see the dynamics is actually quite different between these two cells. So then we tried to build a model of these cells, and let me actually already say that we use the same model that we used for the ground truth model, right? So in this case, this is just an overview. It's not supposed to be very precise looking at details. I'll just highlight some things. So here basically is the same plot as before, right? So how is the optimization doing? And we can see that for both cells, it seems that the intracellular, in this case, we only use two strategy actually. So we use the one with patch clamp alone and the one that uses all the electrodes because it turned out from the ground truth evaluation to be one of the best, right? And also these models were quite large, so it took a long time to simulate. So we said, okay, let's only use this one. In both cases, we see that the intracellularly, it seems that the patch clamp approach, so the SOM approach is better than using extracellular, but extracellularly is the opposite, right? So it's not exactly the same as the simulated ground truth model. But one thing I want you to notice is that the scores here are much larger than the ground truth model, more than double, meaning that overall, we're not getting a very good model out of it, right? And you can also see it from the trace. I mean, you have more or less the same number of spikes and so on, but the differences here are much larger than when we use the ground truth model. Extracellularly, we see qualitatively the same distribution, but also here we clearly have some differences which are a bit hard to see, but you might see, yeah, it's a bit too small. But I mean, maybe let's say the waveforms, when they are biphasic, also the model reproduces a biphasic waveform. When they are triphasic, it's triphasic, but there are clear differences as well. And so we asked ourselves, why do we have these differences? And I think, or we think, that there are two main sources of errors for this kind of data. The first one, and I think that's the one that is actually affecting the results the most, is that there is a model mismatch. So basically, we're using a model from a layer 5 pyramidal cell. So it's a cell that sits on a specific layer. It has a very long apical dendrite. It has a certain kind of dynamics and of distribution of mechanisms and ion channels. We're using a model built on this cell to fit a neuron that it's actually a cultured neuron from an embryonic culture. And, even just from the morphology, it doesn't really look like a pyramidal cell, right? So there might be a model mismatch. So the model itself cannot reproduce that behavior because maybe we are missing some ion channels, or the distribution of these ion channels is not well, let's say, defined. The problem is that there are not many models out there built on cultured neurons, and this was not the point of this project. It was mainly to construct and validate the approach and basically show as a proof of concept that this could also be applied on experimental data. So, but this is definitely an error. And the other problem could be in the extracellular modeling. So the extracellular modeling does make some assumptions on basically how these extracellular signals are generated. And the other thing that could actually impact what we see is that in order to simulate these extracellular signals, you need a very precise three-dimensional information of the relative position between the cell and each of its neurite and the electrons. So if we have some small misalignments in the registration between the imaging and the electrodes, this could result in some mismatches in what we actually observe, right? And then also this modeling framework has some assumptions like of a linear, homogeneous, isotropic, and infinite medium, which might not be perfectly met in our case, but there might be some adjustments that we have to make in order to provide like a better fit in this case. But anyways, again, this was just a proof of concept to show that it is possible to acquire this kind of multimodal data and to apply the pipeline that we developed. Now, we used extracellular signals from HD microelectrode arrays and patch clamp. And we all know patch clamp is a complicated technique, very time consuming. Maybe you sit there for hours and you have data from 1 or 2 neurons, so it's very low yield. And the question could arise is, can we actually build multi-compartment models without patch clamp, right? This would actually, if you think about it, it would require less human, let's say, intervention. It could be more automatized, right? It would be easier in the long run, maybe higher yield. We could build more models, acquire more data, and so on. So we just explored one solution, but the range of possible modalities to record and stimulate neuronal activity is wide, right? So one thing that I think is very promising, maybe we're not there yet, but maybe in the years to come, we're going to get better at this technique, is using genetically encoded voltage indicators. So they are genetically encoded dyes. So they're actually protein that can be expressed also in subpopulation of neurons. They can be expressed sparsely as well, and they actually fluoresce as a proxy of the membrane potential, right? So if you image this kind of neuron and if you this kind of signals, and if you have a high enough signal to noise ratio, you could actually record for one specific neuron a proxy of the membrane potential across its entire morphology, right? So this could be like, let's say, an alternative approach and less invasive and more like high yield approach with respect to patch clamp. And since they're also bright sensors, you could use them also to perform the morphology reconstruction, right? So this could be another way that you say, oh no, but we need patch clamp to reconstruct the neuron. If these are bright enough, you could actually reconstruct the morphology from there. And we could also pair these with HDMEAs because they also have the possibility to stimulate, right? So instead of stimulating intracellularly, we could stimulate from these extracellular electrodes. Of course, you don't have exactly the same precision as intracellular stimulation, but you can have like proxies of sustained stimuli. For example, if you stimulate very short pulses with small amplitude, they kind of build up the potential, so they kind of resemble sustained stimuli. And the nice thing is that you could stimulate from different neurites, right? You could say, okay, this now I stimulate from the soma, but you could stimulate from the dendrite, right? You could stimulate from the axonal initial signal, from the axon. So you could have like a large variety of protocols that excite the cell in different ways. And so this could be something to try out next, let's say in this multimodal view of fitting multi-compartment models. And finally, this is more of an overview. So why would we build this kind of models, right? Of course, models in general are a tool that can be used by scientists to test hypotheses that cannot be tested experimentally, or to come up with new possible questions that we could design an experiment for. But I think that these kind of models are also interesting when it comes to studying neurological diseases, right? I don't think many people are thinking of models in this direction, but imagine that we have a very good technique, which could be a multimodal fitting technique to construct models of cells, even from cultured neurons, right? Now we have very strong cell engineering techniques that allow us to build cell cultures, patient-specific, also from specific diseases. So we could actually build models from healthy subjects or from subjects with a certain specific disease like Parkinson's or ALS. And then we could use these models to build larger network simulations and see what's the emergent network dynamics, how does it change in disease, right? So this in-silico evaluation could really be like a new step in between like in vitro and in vivo that could be more approachable. Of course, it requires effort in like building these models and making sure they are validated, but they could really be used also in this direction as like to support these other in vitro, which is probably most of the times is too simple of models, and in vivo, which most of the times is too complicated of a model, right? So we could have this middle ground in silico evaluation also towards understanding and possibly, I don't know, for drug screening or for treatment of neurological diseases. So to conclude in this seminar, I try to give an overview of what we mean with multi-compartment models, what are the current limitations of the way we construct these models, and then how we proposed and evaluated a solution that is actually based on combining multiple recording strategies and modalities in order to get a richer data set on which we can construct the model. And finally, I presented some experimental results and also highlighted some limitations that are still there and we need to address in order to get good models from these kind of experiments. So with this, I would like to thank you for the attention. Thanks also to the remote participants. And if you have any questions, I'm happy to reply. Thank you.
Okay, thank you, Alessio, for the nice talk. So yeah, I mean, also for the remote people, if there's any questions, I mean, you can put them in the chat or just unmute your microphone if you can, or I mean, raise your hand. Are there any questions from here? Yeah.
So I haven't fit exponential segments like I haven't modeled them. So when you model them or like use the parameters to fit the model, are you using different kind of channels, like, or you are using the same, like we are using the same channels, but in different proportions? Perhaps that may cause the difference in the fit.
That's a good question. So we actually use both different family of channels, and also I think it was mainly Michael who designed this model. We had like specific bounds that were optimized to actually reproduce this kind of dynamics. So it's both the family and also the bounds for these conductances were actually fine-tuned to reproduce this kind of dynamics. But of course, within these bounds, they were still optimized.
And one more thing is Alan database for this cultured neurons. I was checking in Neuromorpho, some databases. Are these all cultured neurons?
I mean, the one from the Allen Institute, they're all for mice or humans.
Mm-hmm.
So I think we are maybe the first ones who tried to build models from cultured neurons. Because I mean, we were discussing also among us, saying, okay, what's really the point of constructing a model of a cultured neuron, right? It's not really these cultures are not a good model of what happens in vivo, right? So slices would be better. But then I think that if you think of cell cultures with their capabilities, as I tried to explain with the last slide, right? With the capability of actually modeling specific diseases, right? It could be powerful to have a way to build accurate models, even of cultured neurons. So they're not there yet. We share the data, and also the morphologies are shared in a GitHub repo. So yeah, we're going to probably put the paper on bioRxiv in a few days.
Thanks.
Thank you.
I mean, related to this, actually, I had also another question. Like, I mean, to avoid using cultures, like, I mean, I assume it would be very difficult, but you think like it's possible to use some in vivo probes or something or whatever to also get like good signals that we could use? I mean,
yeah, I mean, possible, yes, complicated, yes. But I think a middle solution could be, I mean, most of these cell models are built from slices, right? So let's say we agree that slices is a good model, like to build this kind of multi-compartment models. So one thing that you could do, this could be applied to slices as well. And the main problem is that when you place a slice, like you have here on the chip, right? The slice is a little bit thick, right? It needs to be at least 200-300 micrometers thick. Otherwise, it's too thin to have any meaningful activity. But then, in order to record the neurons extracellularly, they need to be close to the electrodes. So basically, when you put a slice on these devices, you are mainly recording the neurons that are sitting at the bottom of the slice, which are the neurons that are harder to patch, right? Because you cannot see that far. So one possible solution to this could be either to wait a few weeks for the slice to kind of culture the slice. So these are called organotypic slices. So you kind of wait for a couple of weeks and the slice in time it flattens, but it keeps all the richness of neurons of different cell types of the slice. And the other solution could actually be to use the extracellular electrodes as like a guidance system for your pipette. So if you think about it, it's sort of like a GPS, right? If with the pipette, as you insert it in the slice, you start sending some small pulses, you could use the extracellular readout to triangulate the position of the pipette, right? So you could try to use it as guidance system. There are some approaches there, like they could say, oh, we can do fully automatic patch clamp with this solution. I think it still needs some work, but it's definitely a potential approach. And in vivo, very complicated. I mean, there are some data sets that have been acquired, for example, using neuropixels data and patching cells, or at least having juxtacellular recordings of cells close by. I think the main caveat there would be to then do the reconstruction and to know the exact relative position of the probe with respect to the cell, because then either you do it live, in a sense, you have a window open and then you have like a 2-photon microscope and you can scan through the tissue, but we're adding more and more technology there. I mean, in principle, feasible, in practice, very hard.
Okay, thank you.